Okay. So the types of tetrasporic embryo sac. In this type of embryo sac, there are several variations. In several cases, 16 nuclei are formed with the result of 2 divisions after megasporogenesis. So they are further subdivided into 7 types. So in that first one is the Adaxa, Plumbago, Pinia, Peperomia, Drusa, Megaspore, that Fritillaria, Plumbagella, and Chrysanthemum. So we are observing one by one in just to follow this. This is the Adaxa type. So in the Adaxa, just wait. Just I'll show the only single diagram. Adaxa, this type of development of embryo sac was described for the first time by Johnson. Johnson in Adaxa mastillica. So, and later by the Lagerberg, and it is also in this type, the 4 megaspore nuclei are divided to form the 8 nuclei. So, which form a normal type of the 8 nucleate embryo sac. Just you can observe here, just 1 and second division, 1 to 2, 2 to 4, 4 to 8 nuclei are formed. So it has 8 nuclei which are formed by the mitotic division of the 4 haploid nuclei of the cynomegaspore. Okay, so the arrangement of the 8 nuclei in the embryo sac is the same as in the polygonum type. Okay, so the example Adaxa, Sambucus, Almus, Tulipa, and Erythronium. So these all are the examples of the Adaxa type. So this type of development of embryo sac first described by Johnson in Adaxa masculatia. Okay, so in this almost in the 4 megaspore nuclei, that means the development of embryo sac take part by the 4 nuclei. So that's why it is called a tetrasporic embryo sac. Okay, so in this type, the 4 megaspore nuclei divide to form the 8 nuclei, which form a normal type of the 8 nucleate embryo sac. So it is very common. Normal egg apparatus, 3 antipodal cells, and 2 polar nuclei. Okay, so this type of embryo sac is regular in Adaxa and Sambucus. It is also occur in some species of the Almus, Tulipa, and Erythronium. So this is very commonest type of the among the tetrasporic embryo sac. And the second one is the Plumbago type. So this type of embryo sac has been reported in Plumbago capensis. Which plant? Plumbago capensis. Here the 4 megaspore nuclei, they are arranging crosswise and divide once to form the 8 nuclei in 4 pairs. Just you can observe here, all that 1 will divide to form the 2, 2 will divide to 4. So 4 are arranged in the 4 places of the embryo. So micropylar, collagel, and 2 adjacent or peripheral portion of that. So that means here the 4 megaspore nuclei arrange crosswise and they will divide once to form the 8 nuclei in 4 pairs. Okay, so 4 will formed as the pairs in the 4 places. So 1 nucleus of the micropylar pair is now cut off to form the lenticular egg cell, and 1 nucleus from each of the 4 pairs, they will approach each other in the center and they will fuse to form a tetraploid secondary nucleus. So almost in the in the center portion only 2x or 2n state we are observe up to here, but in the Plumbago type we are observing the almost tetraploid secondary nucleus. 4 cells are fuse to form the center portion. So that is the center, and they will fuse to form it a tetraploid secondary nucleus. So the remaining 3 nuclei are degenerated. That is the plumbago. So almost in the 3 adjacent cell of the nucleus are degenerated. Only the center portion will functional. So that is also the nucleus from each of the 4 pairs. They will approach each other in the center, and they will form to tetraploid secondary nucleus, and the remaining 3 nuclei are the degenerated. Okay, so the plumbago type of development occurs not only in the other species of the genus plumbago and some other, but also in 2 other genera of the Plumbaginaceae and Ceratostigma. So which plants? So the family is the Plumbaginaceae. For example, Ceratostigma, and other one is the Vogelia. So Vogelia also some other type. This type is not known in any other families except to Plumbaginaceae family only. Okay, so that's why the name is just to put that plumbago type. This is the some other case, some exceptional case of the tetrasporic embryo sac. So character by the absence of synergids and antipodals. Out of 4 haploid, they can one migrate to the micropylar end, one at each calajal end, and 2 at the lateral sides. 4 sides. So each nuclei divides again and formed 4 groups of the 2 nuclei. So each nuclei almost in the one of the nucleus from each group, they can move to the center portion of the cell, and they will form the 4 polar nucleus. So the remaining nucleus at the micropylar is cut off by a membrane, and they will form the egg cell. There are no synergids. So the other 3 nuclei usually disappear or disintegrated, but occasionally they may be cut off by membranes and appear as the accessory egg cells. So for example, Plumbaginaceae family. And third type is the Pinia type. So in the Pinia type, so in this type, the 16 nuclei, they are lie in the 4 distinct quarters. So just you can observe here diagram. The 4 haploid nuclei of the Cenomegaspore undergo the 2 successive mitotic divisions. They can forming the 16 nuclei. Okay, so these nuclei, they can arrange themselves in 4 groups of the 4 each. So in that, one at the micropylar end, one at the calajal end, and one each on the 2 lateral sides. Okay, so same as per plumbago. So this is up to the formation of the 3rd divisions. So now 1 nucleus from each group, they can migrate to the center portion, and these fuse, they can form the polar nuclei. Okay, so that means the 4 cells, the nucleus from each group migrate to the center, and these 4 cells form the polar nuclei. So the 3 nuclei at the micropylar end are cut off by membranes, and they'll form the egg apparatus. So the remaining 3 groups of nuclei, that means in the 1 calajal and 2 lateral sides. Okay, they can degenerate it at maturity condition. So the highly polyploid, that means the 4 or 5 primary endosperm nucleus is formed after double fertilization. For example, family Piniaceae, Malpighiaceae, and Euphorbiaceae. Okay, so this is the Pinia type. That means the 16 nuclei lie in 4 distinct quarters, which are arranged crosswise, 1 at each end of the embryo sac and 2 at the sides. So now 3 nuclei of each quarter, they will become cut off as cells, while the 4th one remains free and move towards the center portion. That means thus there are 4 triads and 4 polar nuclei are formed in that complete embryo. So the egg cell of the micropylar triad alone is functional. Such embryo sac have found in many membranes of the Malpighiaceae. Okay, so the family Pineaceae, Malpighiaceae, and Euphorbiaceae are the best example of the Pinea. And the next one is the Peperomia type. So in the Peperomia type, the egg apparatus of Peperomia type is characterized by a single synergids. That means in this subtype, each of the 4 megaspore nuclei divides twice and forming the 16 nuclei, which are uniformly distributed at the periphery of the embryo sac. So that means the 2 nuclei at the micropylar end, they'll form an egg apparatus and synergids also. And 8 are fused to form the secondary nucleus, and the remaining 6 nuclei are cut off at the periphery of the embryo sac. Just you can observe here 1 to 2, 2 to 4, 4 are arranging in the upper, lower, and 2 lateral sides. So it will undergo the other divisions. They can form the 16 cells. Okay, the forming the 16 nuclei, which are uniformly distributed at the complete 4 sides. In this 2 nuclei at the micropylar end, they will form the egg apparatus. Only these 2 are the egg apparatus and some synergids also. And remaining 8 are fused and form the secondary nucleus. Okay, the center portion 8 nucleate stage. That is the all that 8 are fused to form the secondary nucleus, and the remaining 6 they are completely arranging. They are cut off at the periphery of the embryo sac. That is the lower portion arrangement of the embryo sac. So out of 16, 8 is the center portion, 16 is the lower portion, and 2 is the upper portion. Okay, so out of 16 they are arranging and differ in the center portion. So that is the Peperomia type. So the best example is the Peperomia and Gunnera. This is the Peperomia, and next one is the Drusa type. So in the Drusa type, in this type, a 16 nucleate embryo sac was recorded in Drusa appositifolia. The family is the Umbelliferae. Which family? Umbelliferae family. So here, when the meiotic divisions are over, 3 of the megaspore nuclei pass down to the basal end of the embryo sac, and only 1 remains at the micropylar end. This is followed by 2 successive divisions. They can forming the 4 micropylar nuclei and 12 antipodals. Okay, so the 4 micropylar nuclei, they'll give rise to the egg apparatus and upper polar nucleus, and the 12 collageal nuclei to form a lower polar nucleus and 11 antipodal cells. So this is some other exceptional. This type has been recorded in Malotus and some other Crucianella, Rubia, Almus, and few other plants also. They are arranging this type of the complete development. Okay, so the Drusa means 16 nucleate embryo sac. It was recorded in the Drusa appositifolia. It belongs to the family Umbelliferae. Okay, Umbelliferae or Apiaceae. In this type of embryo sac is characterized by large number of antipodals. The antipodal cells are a group of antipodal cells are presenting. They are the completely. So you can observe this is the complete 12 cells. Okay, antipodal cells, and in the mature embryo sac, 3 nuclei form the egg apparatus. Okay, the lower portion, and 2 act as the polar nuclei in the center portion, bipolar nuclei, and the remaining 11 nuclei are cut off by membrane and they are formed the antipodal cells. Okay, out of 12, 1 is the center portion, remaining 11 cells are act as the antipodal cells. The number and organization of nuclei, it may vary due to the irregularity in the divisions. That is the drusa type. So the example is the drusa, rubia, chrysanthemum, almus, and malotus. Some of the crucianella. These all are the other plants of the tetrasporic type of the embryo. And other one is the fritillaria type. So in this fritillaria, this type of development of embryo sac has been reported in many angiosperms, including lilium. So in fritillaria and lilium, the behavior of the 4 megaspore nuclei is very peculiar. So it's very different. Here, 3 of the megaspore nuclei go to the collagen end, and the 4th one goes to the micropylar end. So the 3 collagen nuclei come to lie very close to each other. During the next stage, the micropylar nucleus divides normally, but the 3 collagen nuclei fuse together, forming a triploid. That means 3 end state nucleus. So which then divides mitotically. It will undergo the forming of 2 triploid nucleus at the collagen end. The close of the division, there are 2 haploid nuclei at the micropylar end and 2 triploid nuclei at the collagen end. So that at the close of the division, there are 2 haploid nuclei at the micropylar end and 2 triploid nuclei at the collagen end. So this is some other differentiation. So whatever it may be, the 4 haploid nuclei of the cenomegaspore, it will arrange themselves in 2 groups. So almost we upper and lower groups. 3 at the collagen end in the form of the triploid nucleus and 1 haploid at the micropylar end. Just micropylar and collagen end arrangement. So in this, the triploid collagen as well as the haploid micropylar nucleus, both are undergo the 2 mitotic divisions, and as a result, 4 triploid nuclei are formed at the collagen end and 4 haploids at the micropylar end. So just I'll show only complete circles in mature embryo. In mature embryo sac, 3 haploid nuclei organized into egg apparatus, 3 triploid into antipodals, and remaining 1 haploid and 1 triploid nuclei move to the center where they fuse to form a tetraploid polar nucleus. So for example, fritillaria, lilium, piper, and gaillardia. So these all are the example. It is very peculiar type of development of the embryo sac. So it is of particular interest because it is found in wide range angiospermic genera, including lilium. So which family formerly was known as the lilium type. So the stages of the development of embryo sac in lilium and fritillaria was fully understood, just like after the investigations. Okay. So in normal cases, all the 4 megaspore nuclei are the same size, but in some plants, the micropylar nucleus becomes large and the other 3 nuclei remaining very small. Just to some, they are arranging as very minute size of the haploids. All are formed in this. So it the tulipa plant, the 3 collagen megaspore nuclei divide abnormally, in which all the telophase chromosome groups are included in a very common membrane, so that the mature embryo sac becomes the some 5 nucleate stage. So for example, all these fritillaria and lilium, piper, and gaillardia, all these example of this fritillaria type. Okay. And next one is the plumbagella type. So this type of development of embryo sac has been reported only in Plumbagella micrantha. Which plant? Plumbagella micrantha. So in this type, the 4 megaspore nuclei take up, it's arranging as 1 plus 3 arrangements. So 1 nucleus at the micropylar end and 3 nucleus at the collagen end. The 3 nuclei of the collagen end fuse together to forming a single triploid nucleus. So now the developing embryo sac is in binucleate stage. Both the nuclei divide once mitotically and forming a second 4 nucleate stage. So that means the 2 micropylar haploids nuclei and 2 collagen triploid nucleus, they are formed in that way. So there are no further divisions. The nucleus situated near the micropylar end, it will organizes into the egg cell. So the triploid nucleus of the collagen end forms the single antipodal cells and the remaining 2 nuclei. So that means one haploid and the other triploid fuse to form a tetraploid secondary nucleus in the center portion. Just to, in the Plumbagella type, the initial development is similar to the fritillaria, same. Okay, that means a triploid nucleus is formed at the collagen end and haploid at the micropylar end. So this look in the 3 and upper is the 1. So it is haploid at the micropylar, 3 at the collagen end. So each of these nuclei undergo a single mitotic division to form the 2 groups of the 2 nuclei each. That means 1 will get 2, 3 will get 9. Okay. So 1 triploid nuclei form the collagen end and 1 haploid nucleus from the micropylar end fuse at the center portion and form tetraploid polar nucleus. Okay. So 1 haploid nucleus at the micropylar end, it will forms the egg cell, and 1 triploid nucleus at the collagen end and form the single antipodal cells. Okay. In this, there is no synergids and no other cells. This is the Plumbagella type. Okay. So the 7 types, almost we are discussing some Adaxa type, Plumbago type, Pinia type, Peperomia type, Drusa type, and Fritillaria type, Plumbagella type, and Chrysanthemum. So whatever Adaxa, Plumbago, Pinia, Peperomia, Drusa, Fritillaria, Plumbagella. These all are the only the main 7 types of the tetrasporic embryo sac. Okay. So this is just, I'll show the one schematic.
Thank you very much, madam. So
this is monosporic, bisporic, tetrasporic. Okay, sir. Okay.
Important for CPGET.
Sir, your voice is not
clearly audible. Okay. So embryo sac నుండి డెఫినెట్ గా మనకి 2 క్వశ్చన్స్ వస్తాయి. ఎట్లా వస్తాయి అంటే ఎగ్జాంపుల్ ముఖ్యంగా ఆంటీపోడల్ సెల్స్ నంబర్ కాంపిటేటివ్ ఎనీ కాంపిటేటివ్ ఎగ్జామ్స్ నాట్ ఓన్లీ ది TS set, AP sets and CA asking about how many antipodals in Drusa type or the Peperomia type or Pineal type to tetrasporic embryo sac or the bisporic embryo sac. So monosporic embryo sac best example ఏంటి అని అడుగుతాడు. How many nucleus are present, how many cells are present in the embryo sac అని అడుగుతాడు. Common embryo sac. అంతేకాకుండా ఒక డ్రూసాలో ఆంటిపోడల్ అడుగుతాడు. అంటే అది ట్రిప్లాయిడ్ కండిషన్ లో ఉంటుందా, డిప్లాయిడ్ కండిషన్ ఉంటుందా, అప్లైడ్ కండిషన్స్ ఉంటాయా అని అడుగుతారు. అంటే యాంటీపోడల్ సెల్స్ కానీ ఎట్ ద సేమ్ టైం మనకు సెంట్రల్ సెల్ కానీ లేదా వీటికి సంబంధించిన అవకాశం ఉంటుంది కాబట్టి ఎగ్జాంపుల్ చెప్పండి, లేదా పీనియల్ టైప్ కి ఎగ్జాంపుల్ చెప్పండి, లేదా పెపరమీ టైప్ కి ఎగ్జాంపుల్ చెప్పండి. మోనోస్పోరిక్ ఎంబ్రియోసాక్ లో కండిషన్ లో ఎన్ని న్యూక్లియస్ ఉంటాయి, ఎన్ని సెల్స్ ఉంటాయి అని అడుగుతాడు. ఈ కండిషన్
which one is functional and which one is degenerated, so micropylar or collagen, so this is in common.
మేడం
Sir. Okay, sir, continue.
ఓకే, ఇందులో ఈ విధంగా మనకు అడగడానికి అవకాశం ఉంటుంది కాబట్టి మీరు మేజర్ గా యాంటీపోడల్స్ సంఖ్య, సినర్జిడ్స్, కొన్నిట్లో సినర్జిడ్స్ ఒకటే ఉంటుంది, కొన్నిట్లో 2 ఉంటాయి. యాంటీపోడల్ సంబంధించి జనరల్ అయితే 3 ఉంటాయి, కొన్నిట్లో 12 ఉంటాయి, కొన్నిట్లో 11 ఉంటాయి, కొన్నిట్లో 9 ఉంటాయి, కొన్నిట్లో ఇట్లా వీటికి సంబంధించి యాంటీపోడల్ సంబంధించిన సంఖ్యను కూడా మీరు బైహార్డ్ చేయాల్సి ఉంటుంది. డెఫినెట్ గా ఈ టాపిక్ చాలా చాలా ఇంపార్టెంట్. ఎగ్జామ్ లో మనకు దిగాల్సినటువంటి క్వశ్చన్స్ ఇందుకెళ్ళి ఉంటాయి. ఎంబ్రియాలజీకి సంబంధించి, ప్లాంట్ ఎంబ్రియోసాక్ సంబంధించి. మేడం, is there any other information regarding of this class?
Okay, sir, with this just the types of EMBRYOSAC and almost I'll show the complete 12 diagrammatic schematic division in coming class. Once only one diagram, but not now.
Okay, madam, proceed.
In coming class, we are discussing that only all the types: monosporic, polygonum, inothera, and bisporic, Allium, endomion, and tetrasporic 7. Completely 11 or 12 diagrammatic schematic PPT I'll share in group, or otherwise I'll show in coming class at the last time of the tomorrow's class, end of the class. Okay, sir? Thank you, sir. Thank you very much.
Shall we wind up the session?
Yes, sir. Yes.
Okay,
Please.
thank you very much, madam. So
Thank
it is
you,
the
thank
most
you, sir.
important topic, my dear friends. మీరు బాగా ప్రాక్టీస్ చేయండి. ఏమైనా డౌట్స్ ఉంటే మేడం విల్ క్లారిఫై యువర్ డౌట్స్ నెక్స్ట్ క్లాస్. ఓకే, ఇట్స్ టైమ్ ఫర్ వైండ్ అప్ ద సెషన్. 9:10. సో 9-10 నిమిషాలు అవుతుంది. ఇంత సమయం దాకా మేడం రియల్లీ ద గ్రేట్ ఎఫర్ట్స్ రిగార్డింగ్ ఆఫ్ అవర్ CPGET యంగ్ aspirants. Your valuable time sparing for our young budding botanists. Thank you very much. Definitely they will prove their level best. Our services definitely will going to fruitful. Madam, thank you very much. Tomorrow we will meet.
Okay, sir. Thank you. Thank you, sir. Thank you all the participants and all the aspirants. Thank you, sir.
Okay, madam.